What specific problem does the BrainBits method aim to solve in the context of neural decoding and reconstruction?
The BrainBits method aims to address the issue of disentangling the contributions of generative model priors and the actual neural signal from brain recordings in the context of neural decoding and reconstruction. It introduces a novel metric that measures how reconstruction performance varies as a function of an information bottleneck, allowing researchers to evaluate the extent to which models rely on neural data versus their inherent priors. By optimizing the reconstruction objective while varying the bottleneck size, BrainBits reveals that many models achieve high performance with minimal brain signal, indicating potential over-reliance on strong priors rather than genuine neural information. This method is critical for ensuring that reconstruction techniques genuinely reflect brain activity and contribute to neuroscientific understanding, rather than merely exploiting model biases. Ultimately, BrainBits provides a framework for evaluating and improving the fidelity of neural decoding methods.
How does the proposed BrainBits method work? Please illustrate the algorithm step by step from the initial neural data processing to the final reconstructions.
The BrainBits method begins by taking neural data, specifically fMRI recordings, and mapping it to a smaller dimensional space through a linear transformation, referred to as the bottleneck mapping, G_L. This mapping compresses the brain data into a vector of size, L, which is then used to optimize the reconstruction of images or text. The reconstruction quality is evaluated using various metrics, allowing for the generation of a curve that shows how reconstruction performance varies with different bottleneck sizes. To assess the model's performance, BrainBits computes a method-specific random baseline, which uses synthetic data to reflect the generative prior without any brain input, and a reconstruction ceiling that represents the best possible performance if the target latents were predicted perfectly. Finally, the method analyzes the resulting reconstructions to determine how much information is extracted from the brain and which brain regions contribute most significantly to the decoding process.
Can you explain a simple example of how BrainBits would work with fMRI data and a corresponding image stimulus? Walk through the steps clearly using specific numbers and dimensions outlined in the paper.
To illustrate how BrainBits operates with fMRI data and an image stimulus, consider the brain diffuser method, which uses fMRI data from the natural scenes dataset. For instance, if we start with approximately 14,000 voxels of fMRI data from the visual area, we can apply a bottleneck mapping that compresses this data down to a size of 50 dimensions. This bottleneck retains the essential information needed for reconstruction while discarding extraneous data. When we run the reconstruction process, we find that this 50-dimensional bottleneck achieves about 75% of the original performance as measured by the DreamSim metric, indicating that a significant amount of information can be effectively captured with a much smaller representation. This demonstrates that even with a drastic reduction in dimensionality, the model can still produce high-fidelity reconstructions of the corresponding image stimulus, showcasing the efficiency of the BrainBits framework in evaluating the relationship between neural signals and reconstruction quality.
What are the theoretical or practical benefits of using the BrainBits method compared to existing neural decoding methods? How does it improve our understanding of the information extracted from the brain?
The BrainBits method offers significant theoretical and practical benefits over existing neural decoding methods by providing a novel metric to measure how reconstruction performance varies with an information bottleneck. This allows researchers to disentangle the contributions of the generative model's prior from the actual neural signal, which is crucial for understanding the effectiveness of reconstruction methods. By demonstrating that high-quality reconstructions can be achieved with a small percentage of the full brain data, BrainBits reveals that many models rely heavily on their priors rather than the neural information, thus improving our understanding of the actual information extracted from the brain. Additionally, BrainBits enhances interpretability by identifying which brain areas contribute to decoding as a function of bottleneck size, allowing for targeted investigations into brain function. Overall, it provides a more nuanced evaluation of model performance, encouraging the development of methods that genuinely leverage neural signals for reconstruction.
How did the authors validate or test the effectiveness of the BrainBits methodology? What experimental design, datasets, and metrics were used to ensure the robustness of their findings?
The authors validated the effectiveness of the BrainBits methodology by adapting 3 state-of-the-art reconstruction methods: brain diffusers, Takagi and Nishimoto, and Tang et al. To compute BrainBits, ensuring a comprehensive evaluation across different modalities. They utilized the natural scenes dataset, which contains extensive fMRI recordings paired with visual stimuli, to provide a robust dataset for their analysis. The experimental design involved inserting information bottlenecks into the reconstruction methods and varying the bottleneck sizes while optimizing the original objectives, allowing them to assess how reconstruction quality depended on the brain's signal. They employed standard metrics such as DreamSim, clip cosine similarity (SSIM), and pixel correlation for images, and BERT for language, demonstrating that the BrainBits message remained consistent across different metrics. This approach allowed them to quantify the performance of the models and understand the effective dimensionality of the bottleneck representations, ultimately revealing that models relied on relatively little neural data to achieve high performance.
What specific results were achieved when applying the BrainBits method to the state-of-the-art reconstruction methods? Can you provide quantitative values or insights that highlight its performance?
When applying the BrainBits method to state-of-the-art reconstruction methods, significant results were achieved. For the brain diffuser method, a bottleneck size of 50 dimensions achieved 75%, 95%, 100%, and 89% of the original performance as measured by DreamSim, clip cosine similarity (SSIM), and pixel correlation, respectively. This represents a reduction from approximately 14,000 voxels to just 50 dimensions, indicating a factor of about 300 in performance retention. Similarly, for the Takagi and Nishimoto method, a comparable trend was observed, demonstrating that both methods have high chance performance due to strong priors. In the language reconstruction approach by Tang et al., a bottleneck of size 1000 was sufficient to recover 50% of the performance, averaged across subjects, as measured by BERT, with Blue and Meteor scores at 26% and 20% of the original performance respectively.